Okay. Good morning, everyone. And I'm just trying to mute myself on YouTube, because otherwise I'll listen to myself twice. Welcome, everyone, to another CIPER meeting. Good morning. And today I will be joined by my friend Ricardo Mesquita. Ricardo is a former student from the FMH, where he did his bachelor's. In fact, he worked with us in an exercise and health laboratory, and given his adventurous spirit, he then after pursued his education by doing a master's in biology of physical activity at the University of Jyväskylä in Finland, and afterwards in Perth, Australia, where he won a grant to make his PhD at the University of Edith Cowan. He will be talking today about his recently submitted PhD thesis topic, which is focused on alpha motor neurons. He is currently involved in other projects as well, such as a multi-center project on neuroplasticity after spinal cord injury. Ricardo, it's a pleasure. Welcome back. The prodigal son returns. The floor is yours. Thank you.
Thank you, João, for the very nice introduction. Shall I share my screen? Okay.
Please do.
Is it working well? Yeah, cool. Thank you, João, for the nice introduction. It's a pleasure to be here, and I look forward to sharing some of my PhD findings with you. So the title of this presentation is Motor Neurons Have Amplification Powers. Can we estimate and modulate them in humans? Question mark. So as we all know, when we voluntarily move our body, we need to send signals or commands from our brain to our muscles. And how do we do that? So we should not forget our spinal cord, which extends downward from the bases of our brain. And then this spinal cord is made up of nerve cells that carry messages between our brain and the rest of our body. And now we're just missing one element that connects the spinal cord to the muscles, and that's the spinal motor neurons, which is the main character of my PhD thesis. Here you can see a representation of a motor unit, which is made up of a motor neuron and muscle fibers it innervates. Motor units are the basic building blocks of motor output. And importantly, and contrary to what people thought until the 80s, motor neurons do not just passively transmit the information that comes from the brain. The responsiveness of our motor neurons to the constant neural messages that they receive is influenced by a wide range of neuromodulators. The strongest ones are serotonin and noradrenaline. And these neuromodulators influence the intrinsic properties of the motor neurons, making them much more responsive to the information that they receive from the brain and the periphery. So we could say that motor neurons have amplification powers, because in fact, without this neuromodulation of these neuromodulators here, which make the motor neuron much more excitable, we wouldn't be able to produce more than 40% of what we are used to produce in a maximum voluntary contraction. And the most evident neuromodulatory mechanism is the activation of persistent inward currents, or PICs. So what are these PICs? Going back to basic human neurophysiology, some of the signals that our motor neurons receive from the brain are excitatory and others are inhibitory. And when a certain threshold in the membrane potential of the motor neurons as a nerve cell is reached, our motor neurons fire and an action potential is generated. And motor neurons have special channels and that are voltage gated. And that means that they are activated with excitatory input, with electricity. And these voltage gated channels induce a persistent or ongoing flow of positively charged ions into the cell. As a consequence, it gets easier for our motor neurons to generate an action potential and they fire quicker. So these persistent inner currents, or PICs, are the amplification powers, and they allow a sustained depolarizing current to the motor neurons that we need for both strong contractions, but also prolonged low intensity contractions. So the overall aim of my PhD project was to elucidate how we can estimate and how we can modulate or change the contribution of these persistent inner currents to motor neuron firing. Today, I'll be summarizing the main findings of 3 studies that were included in my PhD thesis. Initially, we tried to optimize a technique, a method which could hypothetically estimate peak activity in humans. So let's start with that. So we know that peak channels are voltage gated. Therefore, we know that they can also be initiated by excitatory 1A sensory fibers in vivo. So these fibers come from the muscle. And if we stimulate them, can we then take a look at persistent inward currents? Possibly. So that's what we try to do, at least, and try to see how we could optimize the activation or depolarization of these sensory fibers. And how did we do that? So we know that 1A afferents are depolarized by the activation of muscle spindles. Maybe some of you have heard about muscle spindles. They are muscle receptors that are sensitive to muscle stretch or lengthening in our muscles. So option 1 to get these sensory fibers firing would be to use vibration, because vibration basically evokes lots and lots of little stretches that activate these muscle spindles. Option 2 would be to do a direct activation of these sensory fibers, and we can do that with electrical stimulation. So in our lab, we use a technique that we combine both options, and we called it VibStim. So this technique starts with 10 seconds of tendon vibration, as you can see here in this real-time demonstration, and we deliver bouts of electrical stimulation through the skin at the same time. And during this trial, the participant is not voluntarily making any force. But you can see that after each electrical stimulation bout, the force doesn't go back to baseline. So there's an ongoing and incremental production of force. And after stopping both the tendon vibration and muscle stimulation, we can see that this force keeps going. It doesn't drop to baseline straight away. So hypothetically, persistent inward currents, or PICs, could be responsible for these magnitudes of involuntary force. So next time you are in a physio session and you're receiving some electrical stimulation in your muscles, remember that the electrical stimulation is not only going to your muscle directly, but it's also activating sensory fibers that transmit signals all the way back to the spinal cord and back again to the muscle, making your muscle contract even more. So in this study, we wanted to systematically evaluate what would be the optimum method of electrical stimulation to get the bigger response possible, and by altering some of the electrical stimulation parameters. So those were current intensity, so how strong the electricity is, pulse width, which is in each single stimulus, how long does it last, and finally the pattern of stimulation, so the number of stimulus and the timing of stimulation. And then we looked at the effect of these electrical stimulation parameters on the amount of force produced in different time windows during the trial. So we had 20 participants, and surprisingly, we found that 6 out of 7 males, but only 5 out of 13 females were responders. I mean, producing a sustained involuntary force like you saw in the video. And this is why we recruited more females, because we aim to analyze a similar number of males and females. So this is how a responder would look like. But an important thing to take into consideration is that this technique does not produce this effect in everyone. In fact, some people, after you're giving the vibration and after you give the electrical stimulation, it goes back to baseline. Although we have some ideas, we're still not sure why some people respond and others don't. And this large inter-individual variability actually has been reported before in other studies that use similar techniques, such as electrical stimulation only. So in those participants who exhibited an involuntary force production, we examined the effect of intensity. So we compared the intensity to evoke 10, 20, or 30% of maximum force. And we found that 20% intensity induced a greater response than 10%, and there was no difference between the 20 and 30% trials. In fact, 8 out of our 20 participants found these 30% trials painful. So they couldn't, not everyone could complete those. And so what probably happened here is that we had a higher number of sensory fibers being recruited from 10 to 20%. And then in 20%, we find a sweet spot, because in 30%, it starts getting discomfortable and maybe even a bit painful, which causes some inhibition in the motor neuron. Now, another thing we looked at was pulse width, and we found, as expected, that wider pulses were evoked greater torque, and possibly explained by sensory axons being more effectively depolarized by wider pulses. In terms of pattern of stimulation, we compared 2-second versus 1-second bouts, and we saw that 2-second bouts were also more beneficial. Possibly the longer duration bouts may have allowed the cumulative excitatory effect of 1A inputs, allowing peaks to grow within each burst. So in conclusion, we found that this combination of parameters was the one that generated more robust responses and to this technique. And though we tested with during tenant vibration, it's also possible that this combination of electrical parameters could also provide an effective stimulus in other settings, such as neuromuscular electrical stimulation based rehabilitation interventions. And if you'd be interested in reading more, this has been published last year in the European Journal of Applied Physiology. So now that we have found what were the best electrical parameters of this technique, which could hypothetically estimate peaks, our next step was to compare this alternative hypothetical way of estimating peaks with a more established method. And we did that by using these two methods during interventions that could decrease in study 2 or increase in study 3 persistent inward currents. So this is the VibStim technique that we just talked about. And the other technique is called paired motor units technique, which is involve a bit more of a laborious analysis. So to understand the paired motor unit technique, we can start by asking whether we can identify when single motor neurons are firing in humans. Yes, that's actually the only nerve cell that we can do that in humans. And how do we do that? So most of you might have seen an electromyographic signal, or EMG, which is a measure of electrical activity of a muscle when it contracts. And can we take a closer look at these signals and identify the firing of individual motor units? Yes, and that process is called decomposition. So because EMG signals are the summation of the action potentials of active motor units contributing to the contraction, and different motor units have different waveforms. So they look different when recorded on the skin in the electrodes. So it's like they have different fingerprints. So we have very intelligent computer algorithms that can look at this signal and decompose it to then see when single nerve cells are firing. We can do that with invasive intramuscular EMG, basically a fine wire electrodes through a needle in the muscle, or alternatively and less invasive, we can use surface high density EMG, which was what I used in my PhD projects. So these grids or matrices, as you can see, have lots of small electrodes, and we can get an EMG signal for each electrode. So imagine these small electrodes as windows, and through each window, we can then look at how the motor units look like from a slightly different angle. And as a result, the computer algorithms can then differentiate motor units in the EMG signal. And here you can see an example of my PhD data in which we could identify 26 different gastrocnemius medialis motor units. So the muscle in our calf that allow us, one of them that allow us to do plantar flexion. And we could identify this during a ramp isometric contraction. And these methods allow us to better understand how we generate force, because the magnitude of muscle force that we produce depends on the number of motor units that are actively contributing to the contraction, but also on how fast each motor unit is firing action potentials. I should say that HD EMG, so high density EMG, the composition cannot be a black box where you insert the global EMG signal and then extract the information straight away about the firing timings. And so it's maybe one day, but not today. So it's a semi-automatic process, and it requires quite a bit of some fine editing and cleaning of the data. But then we're able to track the same motor units across different contractions. Thus, we can then examine the effect of certain interventions on motor unit behavior. For example, these three ramps were done during a control condition and these three during experimental condition. So that's what I did in my PhD. And in terms of motor unit behavior, I tried to estimate the contribution of these amplification powers to the firing of motor units. So to understand how we can estimate PICs, let's think about one scenario. So when we linearly increase and then decrease the net synaptic input to a motor neuron, maybe in more simple wording, let's imagine a scenario in which our brain starts talking to our motor neurons, but not so loud. But then after a few seconds, it starts screaming, and then it goes back to not so loud. So we would expect a linear increase and then decrease of the activity of the motor neuron or firing, right? And do we see this? Absolutely not. Instead, for a linear input like that, we see this crazy pattern of motor neuron firing. And so first we see an acceleration in firing, and the natural consequence of this strong amplification at the start is then an attenuation of the firing. So besides this dual slope pattern at the start, then we see also another phenomenon, which we call it firing hysteresis, meaning that the motor units start firing at a given level of force and stop firing at much lower level of force or synaptic input. So in other words, our motor units starts firing when our brain is screaming quite loud and stops firing when our brain is almost silent. And this difference is believed to be driven by the contribution of PICs, our amplification powers, to the motor neuron. But we can't, in humans, directly estimate these net synaptic inputs. That would mean that we would have to put an electrode into the spinal cord of humans and see how much excitatory drive is coming from the brain and the periphery. So we need to estimate it. And how do we do that? To look at hysteresis, we use the delta F technique that was created 20 years ago, which uses pairs of motor units to estimate persistent in recurrence. Basically, I would be able to estimate the PICs in this specific motor unit here, which I'm calling test unit, if I would know the net synaptic input like we saw in the previous slide. But we cannot do that in humans. So we use the smooth firing rate of a lower threshold motor unit. So lower threshold means that was recruited earlier. And we use this as a surrogate of our synaptic input. And then the difference of the firing from recruitment to the recruitment is our delta F, which means change in frequency. And that's our estimate of PICs. So bigger delta F means bigger amplification happening in the motor unit of interest. So now you can ask, so but which pair do you use to do this? And it's a good question, and we can use many pairs, but we make sure that this control unit is a good surrogate or proxy or estimate of our synatic input. And we do that with a wide range of tight criteria to make sure that it's really reflecting what's happening. And in fact, to decrease the variability of the measure, we also use different control units to estimate the amplification in our test unit, like using different measuring tapes to measure the same thing and then get an average out of it. So in my studies 2 and 3, I use these two different methods to estimate the amplification peaks in different interventions. Hypothetically, these techniques could be supplementary to each other, as one would estimate peaks in the absence of voluntary drive when participants are not telling the muscles to contract, which could be indicated for people, for example, with complete spinal cord injury that cannot move their muscles, with equipment that is available in many laboratories, and it doesn't involve this laborious analysis. However, it's very hypothetical. So we wanted to compare with a more valid method. And this technique has been used for 2 decades, and it has been validated with animal models and simulation studies. So the studies 2 and 3 actually form part of the same large study. And given the extent of the procedures and the results, we thought we would divide it into 2 studies to be more digestible by the scientific community. And we had 4 conditions and that were performed in a randomized order over 2 experimental days. So the participants came in and did first the VibStim technique with 2 conditions, and then the other technique with other 2 conditions. And also the control trials in these studies were always performed before the experimental trials. So here we have the VibStim and here the voluntary one, the paramotor unit technique. And what were those conditions? We had reciprocal inhibition. So basically we know that these amplification powers can basically stop if we give some inhibitory signals to our spinal cord. And we can do that by actually stimulating the sensory nerves of the opposite muscle, the antagonist muscle. So we did that with the common peroneal nerve stimulation. And there was only some preliminary observations in humans that we could see this effect of inhibition on PICs. Another intervention that we used was simply asking our participants to relax. So in whole body relaxation, the lights of the lab were turned off, and we asked the participants to relax all the muscles, close their eyes during 30 seconds before the trial, while listening to a relaxing piano song. So then participants slowly opened their eyes before the protocol started, and they were asked to keep their muscles relaxed in the VibStim technique and to relax all muscles, but the ones that they were contracting in the ramp contractions while the music continued. So as both arousal and whole body muscle activity might influence the release of serotonin and noradrenaline, which we know that increase peaks, by asking someone to relax, maybe we would reduce peaks, but this hypothesis had not been tested. Maybe some of you are aware that by clenching our teeth, we can produce more force. There are several studies that have shown improvement in exercise performance during jaw clenching, but we really don't know the underlying mechanism. So we asked participants to clench their teeth on a plastic mouth guard that was fitted to their jaw by putting in boiling water first. And then we should expect, when we contract our muscle based on animal data, that we have a stronger release of serotonin. And when serotonin is released in our body, it's released to all levels of our spinal cord. So we could expect that by clenching our teeth would increase serotonin in our motor neurons that also innervate our leg muscles. Finally, the 4th condition was mental stress. In mental stress, we ask participants to count down from a random number in intervals of 13 as fast as possible, and a loud sound like a game show was generated when participants gave a wrong answer. So these increased levels of stress in this condition would likely lead to an increase of noradrenaline release, which could lead to bigger peaks. So this is an overview of the 4 conditions of the studies. And what did we find? So we found that during voluntary ramp contractions, our estimates of peaks decreased during reciprocal inhibition and whole body relaxation as expected during the voluntary technique. And then during the non-voluntary technique, during the VibStim, we didn't see significant changes in all variables in reciprocal inhibition, and some variables, but others no, did decrease in whole body relaxation. So we have a lack of consistency between these 2 techniques in this study. So the previous studies showed the decrease in peaks during inhibitory inputs using animal models, and we now demonstrate these effects using multiple human motor units, as we can see here. And furthermore, the observation of reduction of peaks during whole body relaxation for the first time in humans is probably indicative of a release of a decrease of a release of serotonin and noradrenaline to the spinal cord. Why didn't we see the same effect in VibStim? Possibly there was a floor effect. So given that some participants had low magnitude of responses and these interventions had a negative effect, it means that there may be insufficient opportunity for VibStim variables to decrease. Another option or possibility is that maybe VibStim is telling us something else apart from PICs. So that's possible. These findings have just been accepted for publication in the Journal of Physiology some days ago, and they should be online soon. And here you can see a graphical abstract illustrating the new physiological insights that we think that our results suggest. So this negative effect of reciprocal inhibition on persistent inward currents corroborates the hypothesis of the importance of local inhibition to attenuate the powerful and diffuse amplification that we have in our spinal cord. And very speculatively, when a surgeon, for example, performs a task in the operating room, he or she needs inhibition acting on the motor neurons that innervate the hand muscles to allow very precise and fine movements without big amplifications that could lead to unwanted movements. So maybe without inhibition, we wouldn't like to have amplification powers in our spinal cord. And an additional implication is the possibility that non-pharmacological interventions such as electrical stimulation and relaxation could be used to attenuate conditions characterized by motor neurons being hyper-excitable, such as muscle spasms after spinal cord injury, traumatic brain injury, or multiple sclerosis. Finally, regarding the other 2 conditions, we did not see a change in delta F, so in our estimate of PICs in jaw clenching or mental stress during the voluntary ramp contractions, contrary to our hypothesis. And very interestingly, all variables increased, and quite a lot, in jaw clenching during the VibStim technique. And most of them increased during mental stress. So this is a technique where the participants are not voluntarily contracting the muscles. So again, we have a lack of consistency between the 2 techniques. These results are not published yet. They are under preparation. And it's possible, one possibility for this lack of consistency is that we know that based on animal data, we have release of serotonin and noradrenaline when we voluntarily contract our muscles. So by asking participants to doing a voluntary contraction, maybe there's a ceiling effect because it's not possible to increase even more neuromodulation, limiting further amplification. Speculatively. Another possibility is that because participants were doing a ramp contraction and they had more neuromodulation, they actually had to use compensatory mechanisms for inhibition to actually be able to follow the force trace. Maybe it would have been different in a maximal contraction. Another possibility is that all triceps surae muscles or plantar flexors contributed to the contraction, of course, but we only looked at the gastrocnemius medialis. So it's possible that seeing an effect here in the VibStim technique that should look at all muscles and not here has to do with the fact that maybe the other muscles such as soleus had an effect whereas gastrocnemius didn't have. Soleus has a greater contribution of a greater proportion of smaller motor neurons, and we know that smaller motor neurons rely more on these amplification mechanisms, so that could have happened. Finally, maybe VibStim is telling us something else, that it's not PICs. But the effect of jaw clenching was very, very, very evident. So maybe as a suggestion and speculation, people who are undergoing neuromuscular electrical stimulation could benefit from clenching their teeth than just chilling on the phone to enhance adaptations of this type of interventions, ? So to conclude, motor neurons have amplification powers. Yes, we can estimate them in humans. We've been doing it for a few decades with a well-established technique. In my PhD, I tried to see if another technique could also be used. We still cannot rule out the possibility that that technique cannot estimate PICs. And we found robust evidence that inhibition and relaxation can modulate these amplification powers. And maybe things like jaw clenching and mental stress can modulate these amplification powers at rest, but not during voluntary contraction, although this strength of that evidence is not as robust. So I would like to thank all the collaborators and thank you for listening.
Well, thank you very much, Ricardo, for a really thorough explanation of how your PhD actually unfolded. I'm completely astonished to see how far you have actually reached all the way from a few years back ago until now. So thank you very much for this. And now the floor is open. Just a reminder that we're not only broadcasting on Zoom, we're also broadcasting on YouTube, and questions can be done on both platforms. So please. Is he something? Now we wait a little bit. We see if someone actually has questions.
Can you go ahead, João?
Faster. Although it was at the same time as Silvia raising his hand, but I'll give you permission. Thank you.
Oh, I'm sorry about that.
No.
Well, I'm really happy to see Ricardo. It's really a great pleasure to have you here in this, let's say, this symposium from CIPER. And I want to congratulate you from all the way you are developing your career. It's great that you did your master's first in Givascre, and then right now you are working on your PhD program at Eddie Cowan, a very prestigious institution. So just to say hello here from Lisbon, it's nice to see our former students with a successful career. I'm not expertise in this field, but when I was reading your, let's say, your final sentences regarding if you can estimate and modulate motor neurons in humans, I was just remember when I was a BS student here at our faculty. In the very first year, we had 2 or 3 colleagues that they somehow pay a lot of attention to the issue of proprioception. Proprioception, how proprioception to be, proprioception, if I remember well, it was a term that was coined very early in the 20th century by Sherrington, if I remember well. Sherrington was the first scientist to coin this proprioception. But again, I'm not expertise on this very specific field that you develop your studies, but a general question. Because also I remember when I was a volleyball coach, and someone has, let's say, an ankle injury, ankle strain, and in order to have a quick and efficient recovery, I tend to use, let's say, a recovery based with the Friedman table, in which the player was supposed to improve his proprioception in order to avoid, during landing, to use the other leg and to get injury in the other leg. So in order to improve fast recovery. So, but my very general question is, according to your knowledge, according to your studies, how can we frame this issue of the study of, let's say, muscle, spindles, motor neurons, and so on, within the scope of sport skills? Because I remember I was a student, the role of, let's say, to have smooth movements. And some of when I was really young, they told, okay, we need to pay attention to proprioception. So what's your view about that within the scope of sports development, sports skills learning, and so on and so
on? Thank you, Professor. That's a very interesting question. So in terms of trainability of proprioception, that's a very good question, and I'm actually not sure to which extent we can train our proprioceptive signals or our sensory signals to our spinal cord. But in terms of trainability of these amplification powers in the spinal cord, there's no evidence yet published that training can increase these amplifications, but we could speculate that could be the case. And there's some preliminary observations of that after strength training that have been presented but not published yet. So basically, when we contract our muscles, we are constantly releasing serotonin, most likely, and also noradrenaline. So by stimulating that neuromodulation, maybe we can cause some adaptations in the spinal cord and make the motor neurons more responsive to the influence of these neuromodulators. Of course, this is very speculative. If that happens, then that could be beneficial in a few different ways. In one way, it would allow our brain to don't work as hard when we have to do low intensity prolonged contractions. For example, contractions of our core muscles when we do explosive contractions of the agonist muscles. And in terms of the agonist muscles, if we have the motor neurons that innervate those muscles have bigger amplification, it means that we can contract those muscles faster and stronger, and in a more stronger way. And of course, this is all in theory because it hasn't been shown yet.
Yeah, it's great, because I remember that one of my professors would say the muscle should be understood as a second brain. The more that we know about the muscle, the function of the muscle, and also the metabolism that is related with the muscle. It seems that really, I don't know if it's the second or the third or anyway. So, but really a lot of things are going on in the muscles. So, Ricardo, I wish you all the best for your career, and I can see that in the future you'll be back here to Portugal. Okay, all the best for you.
Thank you.
Thank you, Professor. Sylvia, please.
Hi, Ricardo. Thank you so much for your very interesting presentation. I had a few questions popping up on my mind, but I'll start with this first one. Maybe then I'll give the word to Adriano, and then if there are no more questions, I'll ask the other ones. Otherwise, I'll just take all the time for me. Well, being a biomechanist from the human movement assessment area, I was curious. I noticed in the picture where you had the person fixed to the dynamometer, it looked like you had reflective markers on the dynamometer. Did you? Why were they used?
That was simply because another friend of mine was using the equipment at the same time for motion capture. I didn't use it in my study because these were not dynamic contractions.
Okay. So that's out of the way. Then the other question, I hope it's not a dumb one, but why did you feel that you need to, or why did you want to try a new technique? So the VibStim, right? The new technique to estimate peaks. I understood that there was another established method, or it wasn't very good. Or...
Yeah, that's a good question. So we do have a more established method, and maybe I'll just quickly share the screen again. And so just to go to, so we have these two different methods, and the pair motor unit technique has been around for 2 decades now. And in this technique, we need to ask the participants to voluntarily contract their muscles. So one advantage of this technique compared to this one is that we don't need to ask participants to voluntarily contract their muscles, which can be important in people, for example, with spinal cord injury. Also, this analysis is quite laborious, and this analysis is not as laborious. So by doing this technique, you would also be more, allow a bigger widespread of the technique in the community. And also more importantly, although I didn't talk about it today, with this technique, we have seen effects that are suggestive of influence of persistent in recurrence. So for example, after passive stretching, we see these responses going down. Or after holding the antagonist muscle at a longer muscle length that would cause inhibition in the spinal cord, we also see these responses going down, and those effects have also been observed here. So it means that maybe at least in some conditions they are telling us similar things. So yeah, that was the main motivation, because this has not been around, and we wanted to see if we could find more evidence that they are telling us similar things or not.
Okay, thank you. Maybe I missed that, but yes, I see the importance, having worked with cerebral palsy children as well, so that would be very useful.
Yes.
Thank you. I'll pass my word to Adriano.
Thank you, Sylvia.
Okay, let me just take my hand down. Ricardo, first of all, a huge congratulations for your presentation, such an in-depth presentation and so detailed. Before I do my questions, I cannot start without make a personal statement. How happy I am and proud to see how far you get and the path that you are making for yourself. So, Ricardo, the first question that I have to ask you is a curiosity. Why do you think that males are more responsive than females? And this is just a curiosity. A second one, this is also not my area of research, but I saw that you had some experimental conditions that you manipulate environments and that you ask people to self-regulate the capacity to relax or contract. And my question is, do you think that your research could give coaches in the future the ability to manipulate some environments and athletes to the ability to self-regulate to a, let's say, optimal capacity to relax or contract depending on specific sports abilities?
Yeah, thanks, Adriano. It's very nice to see you here. Those are very good questions. So I have no idea why we found more responders in males than females. That was very unexpected and could also have been due to chance. And especially because then, although I didn't say today, but then in the other studies 2 and 3, I actually did an effect of sex between males and females to see if these responses were different, because I had more statistical power to conduct that analysis, and we didn't see any difference. But it was still a quite small number of participants, like around 10 in each group. So that this lack of sex effects on PICs should be corroborated by future studies with more participants. But one thing that it's interesting to think about, and that I could quickly show, is that maybe the effect of males and females is the differences between males and females might be different depending on sex hormones. So we know that for example, GABA, which is our most abundant and most common inhibitory neurotransmitter in our nervous system, is decreased by estrogen and increased by progesterone. And we know that inhibition affects PICs. We also know that serotonin affects PICs, and estrogen increases serotonin, and progesterone has the opposite effect. So an important factor to also consider is the possible effect of sex hormone variation during the menstrual cycle on synaptic inhibition and neuromodulation. So that would have a different effect. For example, if a woman is in a monophasic pill, then you don't see changes in the sex hormones throughout the menstrual cycle compared to someone who has a natural cycle. And in people who have a natural cycle, it's therefore possible that the contribution of PICs is greater in the late follicular phase and smaller in the mid-luteal phase. But this is just very hypothetical and it has not been tested. There's some preliminary recent evidence that showed other neuromuscular variables that were bigger in the late follicular phase and smaller in the mid-luteal phase, and who knows, maybe has to do with persistent recurrence. And regarding your other question, it's very interesting, and I think that controlling the environment or asking athletes to relax, or the opposite, to get stress, it could be an interesting intervention to affect performance. But based on so we can see that relaxation decreases persistent inward currents. That might not be something you want to achieve in most sports performance scenarios, but maybe it could be an intervention, very speculative, that could maybe decrease the likelihood of having muscle cramps, because we don't really know why we have muscle cramps in our muscles, but one possibility could be the involvement of persistent recurrence. So the ability to fully relax maybe could have an effect on that. And Adrian was my tennis coach when I was a kid, and when I was very nervous in tennis tournaments, I used to have lots of muscle cramps. So I wonder if it's because of this increase of neuromodulation in our spinal cord. And then maybe the ability to do the opposite, to make them a bit more active, a bit more stress could be beneficial in some scenarios to increase neuromodulation. But my study that looked at voluntary activity and the effect of mental stress didn't show an effect. So you wouldn't suggest that.
Okay, thank you, Adriano. While taking the opportunity to have a little bit of a chat with Mr. Mesquita, we also have questions from YouTube, and people have 2 questions. Okay, specifically, I will start by Pedro Judis, a fellow colleague as well. It says, awesome presentation, my friend. I would like to know if you I would like to know if you have any data about this non-responding issue according to age. Cheers.
Oh, that's interesting. I don't. But I also haven't looked at it. Well, maybe I have the age of my participants, but they were all very similar. There is 2 recent studies that have shown that older adults have lower persistent in recurrence than younger adults. So maybe there's a reduction of PICs with aging. But that was done with the voluntary technique, where the issue of non-responders is not a problem. But that could indicate that you could find it more difficult if I have an older participant coming to the lab and I do that technique. I would speculate that it would be more difficult to produce those responses. But this is just based on what we know from the other technique, because I don't think anyone has looked at it yet. Yeah, but that's an interesting question.
Thank you, Pedro. Thank you, Ricardo. Finally here from Daniel Carrillo. Are there any suggestions about why participants or certain participants maintain torque during more time after the stimulus? Did you do any profile in terms of exercise level or sports participation?
So for the first question, yes, there are suggestions, and I have many of them that I would be happy to share with you. I'll just quickly share my screen again. Regarding the second question, it's also very interesting, and actually a researcher from our research group is actually looking at that question. Is comparing trained versus untrained participants and see if it has an effect on persistence in recurrence, and if it does, maybe it could also affect the responsiveness to the other technique. So why some people respond and others don't could be due to many different things. So one of them is the vibration propagation. Maybe when we're vibrating in a tendon, we can cause more vibration in some people than other in the muscle. Some people might have muscle spindles that are more sensitive. When we do electrical stimulation, people might have different sensory thresholds of the nerve fibers. Maybe this is another thing that happens in the spinal cord. So a post-activation depression of afferent terminals could be different in different people. When we vibrate, we also cause the polarization of some 1B afferents, which have the opposite effect that could be more pronounced in some people than others. The vibration could also go to the opposite muscle in some people more than others, which would cause the opposite effect. Some people might have more or less levels of serotonin, noradrenaline in the spinal cord. Some people might be more relaxed than others when they receive the electrical stimulation. And that could also cause the differences in response, and others biophysical mechanisms related to the channels in the spinal cord. One thing I can show is that, for example, this is an example of electrical stimulation during vibration, and this is a 20 hertz electrical stimulation, and we can see that between each stimulus, we see where did my laser pointer go? I don't know. we see like these little evoked response. Oh, there you are. Between each stimulus, and that means that we are at least able to get those sensory fibers in those non-responders. So maybe that's not the cause. I have some data which I haven't analyzed yet, that I use accelerometers in both ends of the leg to see if those who don't respond, if they have bigger vibration coming to the opposite side of the leg, this would cause reciprocal inhibition in the spinal cord and cause the opposite effect. So this could be something to take into consideration. So yeah, as you can see, many things that could contribute, and very speculative.
Well, thank you, Danielle. Thank you, Ricardo, once again. Sylvia, one last question so you can close up.
Thank you. Just one quick question. Why did you also choose vibration rather than just stretching? And can it have any effect on EMG readings?
So the vibration, we thought that by using vibration on top of the, sorry, did you ask vibration on top of
Stretching? No,
yeah.
Stretching.
We didn't use stretching in our methods. So we used vibration that causes lots of small stretches in the muscle, so acts as lots of small stretches, and we used vibration on top of neuromuscular stimulation because we thought that would give an additional stimulus to the sensory fibers that come from the muscle, because most studies have looked at involuntary forces during electrical stimulation only, and we thought that using vibration on top of that would cause a bigger afferent excitatory stimulus to the spinal cord than using stimulation alone. And vibration was done at 115 hertz, and I didn't see any artifacts in the EMG signal of the vibration.
Thank you.
Okay, I think this comes to a full circle. Thank you very much, Ricardo, for this nice presentation. Everyone, we'll be back next week. And Ricardo, I also challenge you. I know that you've been here before watching our sessions, but to keep coming back so we can see you from time to time. Thank you very much, everyone, and see you next week.
Bye.
Bye-bye.